各位盖亚科普讲座的线上听众，大家早安！很高兴又来到了每月一度的讲座，我是主持人中央大学网络学习科技研究所施如玲。今天我们很荣幸邀请到刘斌教授，他是新加坡国立大学教务长讲座教授，他在学术领域是杰出顶尖的研究员。我发现啊，他有4座院士的这个 title 这样。他是美国国家工程院的院士，是新加坡国家工程院的院士，是新加坡国家科学院的院士，以及亚太材料科学院院士，还有不要忘记他还有英国皇家化学会的会士的这样的身份。那刘教授毕业于南京大学，那后来呢在新加坡国立大学取得他的博士学位。毕业之后，他在加州大学 Santa Barbara 进行博士后的研究，也在2005年加入了新加坡国立大学。后来在2014年，很快的就成为了院长讲座教授，也在2017年成为教务长讲座教授。那刘教授的研究呢，聚焦在有机奈米材料，应用在能源以及生物的医学。那听起来是离我非常遥远的一个专业哈。那但是呢，他在这方面非常的专精，所以他在2014年到2021年的这个期间呢，就被选为是世界最具影响力的科学头脑。他在材料科学以及化学呢是 top 1% 哦，也就是全世界吧哈，全世界的在这个领域里面呢是前1%的被高度引用的这个研究者。那刘教授获得非常多的殊荣，这几年呢，也包括了美国化学学会的奈米讲师奖、英国皇家化学会的100年奖，以及 Caliber 年轻研究员奖。我真的是非常非常的佩服，不知道刘斌教授怎么做到的哈。那我们就真的是非常期待刘斌教授呢，今天来到我们这个盖亚讲座，其实也符合他基于这个培育下一代研究，以及鼓励女性投入科学工程以及奈米科学领域发展的这样的一个工作，那也给我们做一个模范，然后让我们今天的听众呢知道您怎么来到了这样的一个领域，而您的领域又是在做些什么。那今天的讲座呢是要谈材料与生物医学的应用，那我们呢把专业留给讲者，那我就不继续多说，让我们来邀请刘教授开始今天的讲座。聚集诱导发光材料及生物医学应用，刘教授。
谢谢施老师，各位老师同学，大家早上好。Good morning, everyone. It's really my great pleasure to have this opportunity to share with you some of our recent research. We focus on a special type of luminescent materials with aggregation-induced emission. So my research group is very much interested in luminescent materials for biomedical applications. When we look at the cell imaging, it's very nice, very pleasant to work with. So the key of high quality imaging is not only dependent on the equipment, but of equal importance, it relies on the quality of the imaging reagents. And specifically, we talk about fluorescence imaging reagents today. Now, fluorescence reagents can be broadly classified into 2 different types. One type is the aggregation cause quenching, which means these molecules are highly luminescent by themselves when they are very well dissolved in a solvent. But once you induce the aggregate formation, the fluorescence will be quenched. That leads to very low emission, and this phenomenon is not good for biomedical applications. So what we're interested in is a different type of molecules with aggregation-induced emission. These molecules are not emissive as molecular species, but once you induce the aggregate formation, you will observe intensified luminescence. So this phenomenon actually was discovered by Ben Zhongtang from Hong Kong UST, and we really take advantage of this type of materials for biomedical applications. So one of the typical example is the tetraphenylethylene. So these molecules, when they are very well dissolved in organic solvents such as tetrahydrofuran, they're not emissive. But once they come together to form the aggregates, they become very emissive. And that is the optical feature we're very much interested in to use them for biomedical applications. Now, if we look at the molecular level, so this is one of the example of the ACQ molecule. They usually have very planar structure, and therefore, when the molecules come in together, they tend to have strong pi-pi interaction. They stack to each other, and therefore, the excited state is often quenched. And that's why this molecule dissolves very well in tetrahydrofuran to give out greenish emission. But once we add water into this molecule, you will observe that the fluorescence actually is quenched until almost completely darkness. Now, on the other hand, when we look at the AIE molecules, they're not emissive as molecular species because they have these rotor structures and they keep on rotating or vibration that consumes the excited state energy. But once we're able to induce the aggregates formation, these molecules start to stack with each other. And this intermolecular interaction is just about sufficient to restrict the molecular motions, but it prevents the strong pi-pi stacking among or between the molecules so that we're able to observe very strong luminescence. Now, if we open any of the biological catalog, so here I use the organelle staining dye as an example. So many of these dyes are available in the DMSO stock solution. They're highly luminescent. But by the time we use them, we dilute the stock solution with cell culture media or add in buffer water into the DMSO solution. And what we find out is that the fluorescence is quenched and almost completely quenched by the moment we apply them into the cell imaging experiments. So this actually forces us to use extremely diluted solution so that we try to prevent this aggregation called quenching. And this causes another problem, which is photo bleaching, because we can only use very limited amount of molecules for cell imaging. So what we propose here is the AIE molecules. They behave completely opposite to the traditional fluorophores. AIE molecules are not emissive in the MSO solution, but once we add water or cell culture media, we can induce the bright luminescence, and that's very much useful for the biomedical applications. And the same phenomenon is also observed in the solid powder state, so that the AIE molecule shows very high luminescence in the films or in the powders, which make them very important for light-emitting devices as well as biomedical applications. While traditional fluorophores, very often, they show the aggregation cause quenching, and then you lost optical properties in the powder state or in the film states. That's an issue that we have to overcome to explore their applications in many areas. Now, from the molecular design perspective, so this slide shows 5 examples, which are the typical design examples, but it's just few among many that have been reported in the literature. So all of these molecules have something in common. They have these rotor structures with a lot of binding rings. They can move freely in solution as molecular species. So these molecules can be designed to have emission that varies from UV visible to near infrared that will be used for different kind of biomedical applications. So these molecules can be designed to be not emissive or with extremely weak emission as molecular species. But once they are induced to form the nanoparticles or form the films, they become highly emissive. So over the past 2 years, because of the COVID, we actually had a lab shut down for about 2-3 months time. So that was a moment I started to collaborate with my colleagues Wang Xiaolan on machine learning for the AIE molecules. So the idea is when we design the molecules, we often do not know whether they will eventually be an AIE or ACQ molecules, so that we often rely on the trial and error to find out which molecule actually has the desirable properties as we want. But because we don't know, we have to do the synthesis first And then after, let's say, 3-6 months, we obtain a molecule, and then we test, and then we know the results. And we feel that this strategy is common, but it's not very effective in terms of advancing the materials innovation. So together with my colleague, we collected 356 molecules from the literature, among which about 220 ACQ, about 130 AIE. And we rely on the systems and machine learning strategy actually to convert the chemical structure into the code that computer can understand and can learn. And we further actually utilize a lot of existing machine learning strategy. So we didn't really innovate the methodology for machine learning. We just used the machine learning strategy applied to the materials that we're interested in. So through, I think, 5 to 6 rounds of learning, we are able to come out with software or with a scheme that allows us to predict whether a molecule is AIE or ACQ with more than 93% of the accuracy. And based on this new modeling tool, we actually test different kind of compounds. And we on purposely select structures that are very similar with only maybe 1 or 2 atoms difference. We're trying to use the system to predict whether the molecules is AIE or ACQ, and it turned out to be very effective. So eventually we published the database together with the software. So now if someone is interested in predicting whether a chemical structure is the AIE or ACQ, you can simply click on these 2 links. The first link will help you to convert the chemical structure into a SMILE code. Basically, the computer can understand the code that reflects the chemical structure of your interest. And then the second link is the prediction whether this molecule is AIE or ACQ. It's very simple. Once you click on it, it pops up, and you put in your SMILE link, and then you click on the predict. It will give you the answers of 1 or 0. So if you get a 1, it's the AIE. If you get a 0, it's the ACQ. And in total, we have programmed 24 machine learning methods in the prediction. And then at the end, after you see so many 1 and 0, or only 1, only 0, you will get a final recommendation. If it's 1, most likely your molecule will be AIE. If you get 0, most likely your molecule will be ACQ. So with this software ready, everyone actually can test out the molecular design so that before you do the synthetic work, you already know pretty much with more than 90% of accuracy that your molecule will have the AIE property or your molecule will have the ACQ property. So that allows us to really fast screening a lot of molecules at the design stage, and has successfully improved the molecular design in terms of how we can get the properties as we want. Now, with many AIE molecules in hand, we went forward and conducted 2 types of molecular design, and with a purpose for biomedical applications. So the first type is the molecular probes. We take advantage of the very weak or low emission of AIE molecules as molecular species. On the other hand, we also look into the molecular aggregates. That's how we can bring the AIE molecules into the nanoparticles so that they can be dispersed in the aqueous media for biomedical applications. So this is the first type of the AIE light-up probes. Basically, we design the AIE molecules with the recognition elements, and once it interacts with the specific analytes, the lock and key interaction will restrict the molecular motion to turn on the fluorescence of the AIE molecule. So this molecule can be used for many applications. One example is the cancer cell imaging. So this probe will specifically interact with the protein markers on the cancer cell surface, so that in 5 to 10 minutes, we will be able to spot out a specific cancer cell among many other cell types. And the same design principle has also been utilized to develop all different kind of imaging reagents for cell organelles. So basically, we're providing a biological tool for our biomedical and biological colleagues to use them to visualize a lot of cell organelles and understand the cell behavior and functions. Now, the second type is the reaction-based. So we have the AIE molecular probe. Once it undergoes a chemical or biological reactions, the environment of the AIE molecule will be changed so that it will be able to turn on and off the fluorescence at the indication of a particular reaction that occurs in the biological system. Now, one of the very specific example is to look at cell apoptosis. And during the cell apoptosis, caspase 3 and 7 will be activated. And once the enzyme is activated, it actually can cleave the peptide between the K and D. So that's the biological reaction that we're looking into. So in this case, we design an AIE molecular probe, which has the AIE molecule together with a peptide sequence that responds to a specific enzyme in the cells. We have to make sure that the probe is not emissive as molecular species. We also need to balance the molecular probe to make sure that they are able to enter the cells by themselves and yet remain not emissive even inside of the cells. Only after this specific biological reaction, it will be able to cleave the peptide from the vicinity of the AIE molecule, and it will induce the light emission at the indication of enzyme activity that is linked to the apoptosis pathway. So indeed, this is what we observed. Upon incubation of the cells with the probe, along with different time, we observed the light emission that has been intensified, and it takes about 60 to 75 minutes to actually monitor the cell apoptosis process. And this process, usually, if we use traditional biological methods, it usually takes about a day or two to really have a clear indication of the process. So because of this light up probe that allows us to visualize the process, we can use it to actually monitor or predict the treatment of some of the drugs. So here we are using DMSO as the control, and then 3 drugs. We look at the fluorescent turn on with time as the indicator of the effectiveness for the drug to induce the cell apoptosis. So we can see that there are actually intensified fluorescent signals that are observed when the cells were treated with the drugs, but not with the control. And if we look at the intensity profile with the concentration, we're about actually to offer a semi-quantitative signal that is corresponding to the different drug concentration. So this whole process will just take about an hour. So that significantly reduces the time needed for the traditional drug screening in order to understand their effectiveness in inducing the cell apoptosis. Now, after we actually developed and designed a series of molecular probes for sensing imaging applications, so very naturally, we asked ourselves a question: is it possible for us to add more functions into this reporter molecule so that we can transform the original sensing imaging probe into the next level, which is the theranostic probes that not only cover the sensing imaging function, but also incorporate the therapeutic function, because whenever we do the diagnosis, it's for the purpose of treatment. So that's the motivation that drives us to incorporate a lot more functionality into the same molecular design in order to upgrade our molecular probes. Now, one of the examples I would like to share with you today is the organic photosensitizers. So some of you might be familiar with this, and some of you may not. So basically, it's referring to a type of molecules where upon the light illumination, electrons will be excited from the ground state to the excited state, which will undergo the intersystem crossing to the triplet state and react with oxygen to give you the single oxygen species that is very harmful to cells and bacteria, etc. Basically, we can use that as a killing reagent to kill cancer cells and bacteria. Now, for the traditional molecular design, because of the strong non-radiative decay, so that both the fluorescence as well as the single oxygen production yield is not very high. So when these molecules are in the aggregated state, usually we will only have very minimum fluorescence and also significantly reduced single oxygen production rate. Now, what we propose here is to fully take advantage of the AIE molecules, which in the aggregated state, the aggregation-induced molecular aggregation, right? And also because of the special rotor structure that prevents the molecular packing, so that we are able to largely eliminate or remove the non-radiative decay process in the aggregated state. So that allows us to simultaneously enhance the fluorescence process as well as the single oxygen process. Now, for this molecule to be effective, we have to reduce the energy difference between the single state and triplet state so that the intersystem crossing can be effective. So these are the two strategies we introduced to the photosensitizers in order to make a difference to get our photosensitizers highly emissive, as well as being able to produce a large amount of single oxygen at the same time. Now, let me give you very specific examples of how the molecular design is done in order to achieve the goals step by step. So we started with tetraphenylethylene, which is a molecule that does not show obvious photosensitization. And we add in the donor acceptor molecules, and we further add in the pi bridge to enhance the single oxygen production because we're able to change the energy difference between the single state and triplet state. And the next few steps is to improve the light absorption as well as to rate shift the emission so that we can achieve a better penetration depth when the molecules are more emissive in the red and near infrared as compared to that in the UV and visible region. And subsequently, we also look into the polymerization, how we change from a small molecule to a longer chain polymers is able to help us to add in more energy levels in the singlet and triplet state so that we can further modulate intersystem crossing. And very recently, we look into the biodegradable photosensitizers. We wanted to make sure that when we introduce a drug or a therapeutic reagent, once it is done with the treatment, their side effect should be significantly reduced. And one strategy is to make them biodegradable so that they can be biodegraded and clear from the body within a very short period of time. And another strategy to minimize the side effect is to do the in situ synthesis. Means in order to send a drug to the cancer site, instead of sending the drug, we send in two molecular fragments, which are not toxic, but we utilize the microenvironment of the tumor cells, which can induce a chemical reaction that is specific to the tumor, so that it produces the drug or a photosensitizer in situ that shows high selectivity to the cancer cells, but not to the normal cells. And I will talk about that at the end of this presentation. And what we're very excited about is when we compare the photosensitizers that we designed in the laboratory with the state-of-the-art commercial photosensitizer, which is a porphyrin-based molecule, we did observe that our nanoparticles are able to show much higher brightness, as well as the capability to produce stimulated oxygen with the low loading of molecules into the nanoparticle format. So that gave us the opportunity to produce organic nanoparticles that are very bright and with strong capability to produce stimulated oxygens for image-guided surgery and image-guided therapy. Now, again, because of the COVID, we got nothing to do. We're stuck at home. So we also take advantage of the machine learning to predict the organic photosensitizers. So in this study, we actually integrate the DFT molecular simulation with the Bayesian optimization. And we look into about 7 million of compounds where we take about 90 donors and 90 acceptors, about 13 bridges, so that we form a molecular pool of about 7 million compounds. And we use the machine learning in order to predict what is the likelihood for this 7 million compound to produce something effective in terms of the light emission as well as the photosensitization. And after 7 runs of study and prediction, we are able to recommend slightly over 5,000 molecules that shows great potential. And among the 5,000 molecules, we further manually pick up about 10. And here I show 4 of them. So these molecules actually shows quite nice absorption and emission spectra in terms of the Stokes shift, in terms of the molar absorptivity, as well as the emission in the red and near infrared region. But most importantly is that if we compare the performance of these dye molecules with the commercial photosensitizer, we indeed find out the molecules recommended by machine learning are more effective in terms of the stimulated oxygen production per unit time upon shining the same light with the intensity. So they basically behave much more effective as compared to the commercial photosensitizers in producing stimulated oxygen. Now, after we are able to fast screening many molecules and synthesize a lot of very effective photosensitizers, the next question basically is, what can we do about these molecules and what is the difference that we can make? So I want to share with you one example here is how we handle the situation of bacteria that are already invaded into the host cell. And we understand that there are so many antibiotics that are effective in killing all different kinds of bacteria. But many of these antibiotics are not effective if the bacteria is already invaded into the cell. And the reason is that many antibiotics are not really cellular permeable, so that they cannot enter into the cells in order to kill the bacteria. What we wanted to do here is to design the AIE molecules with the terminal amino acids. And we hope this molecule to be so smart that it can penetrate into the cells and find out the bacteria, and then incorporate the molecule into the peptide glycan of the bacteria so that it light up the bacteria, that we can see them. And this will allow us to further conduct the photodynamic treatment to kill the bacteria, and hopefully it will not harm the cell host. So how do we know that this molecule is really able to work with the bacteria and then label the bacteria? So we did a detailed analysis which reveals that after the metabolic engineering between the molecules we design and the bacterial surface, right, we're able to identify this dimer structure from the peptide glycan with the exact molecular weight that indicates our molecule has been incorporated into the terminal of this amino sequence. And we also study the cell imaging and use commercial stain, right? So we know precisely that our red color actually is located on the surface of the bacterial membrane. That is the cross-validation as compared to the mass spectral analysis that proves indeed these molecules are incorporated into the bacteria and they stay on the bacterial surface or membrane. Now, if we conduct a living experiment where we treat the bacteria with our molecular probes, and again, the advantage here is our probe doesn't show any background signal, so that it lights up gradually in the presence of the bacteria of interest. In this case, it takes about 15 to 20 minutes to be able to clearly identify the location of the bacteria. Now, what we care the most is not only how our molecules are able to locate the bacteria, but more importantly, we wanted to make sure we're able to capture the bacteria who are already inside of these cells, right? So in this experiment, we actually treat the cells with the bacteria. And after that, we add in our dye molecule. We hope to be able to capture the bacteria that are inside of the cell. And here shows the cellular imaging. So this is the one big cell. Inside of the cell, there are many red dots. These are the bacteria. Each of the red dots is one bacteria. Bacteria in this case is smaller than the overall cell. So once we're able to visualize them, we know the location of the cells so that now we can actually apply the light to induce the photodynamic treatment. And we can precisely kill this bacteria one by one. And that's why after the light irradiation, the cells inside, the bacteria inside the cells actually will undergoing the dying process in a few minutes or few tens of minutes. We further cared about whether during the process we are offering the selectivity among the bacteria and the cells, right? So here it shows that if we treat the cell that has been invaded by the bacteria with 20 micromole of the molecules that we designed, we are able to kill almost 99% of the bacteria, and yet at the same time, the whole cells is with a very good survival rate, more than 90%. So that shows the selectivity of how we can precisely kill the bacteria inside of the cell and yet still leave the whole cell intact. And that's exactly what we need to achieve in many cases when we are actually infected by the bacteria. Now we further conducted the animal experiments where we look at how effectively this molecule is able to circulate across the whole body of the living animal in order to find the location where there is infection by the bacteria. And it turned out to be quite magic that if we inject the molecule from the tail vein of the mice, it indeed can travel along the whole animal and then locate in the area where there are bacteria that are pre-injected in order to induce this infection. And these are the detailed analysis of the bacterial counting, the distribution of our dye molecules inside of the living system, as well as with the treatment and without the treatment, how the slice of the tissue actually looks very different. So if we don't treat it, many of the cells are dead. Then you see all these voids. If we treat with our molecule, actually you see the tissue that is as fresh as without the inflammation. So that's a clear indication of the importance for the molecular design that really can solve a lot of problems. In this case, is to allow us to detect, visualize, and then kill in one shot. All right, so in the next 20 minutes or so, I will focus more on the nanoparticle side. right? So nanoparticle side is different from the molecular probe side just now, where we take advantage of the molecular species that are not emissive by themselves, and then the light emission is intensified upon recognition or upon a specific biological reaction. Now, for the nanoparticle is the other extreme, where we take advantage of the very high luminescence of the AIE molecules in the aggregated format, so that we wrap them with a block copolymer and bring them into the aqueous media so that they can be used for sensing, imaging, tracking of biological processes, or simply image-guided therapy and surgery. And our strong motivation is to create organic nanoparticles that can benchmark quantum dots in terms of brightness, in terms of biological performance, and yet we're offering a very good biocompatibility that traditional quantum dots cannot offer. And similarly, when quantum dots is dependent on the size to modulate the emission, but AIE nanoparticles is dependent on the fluorophore design to modulate the light emission, which is not really size dependent for organic nanoparticles. Now, the nanoparticle synthesis was based on the self-assembly of block copolymers, where we have a lipid, the SPE, and then the PEG, which is a block copolymer. So this polymer has the hydrophobic side and hydrophilic side. So when this polymer is injected from a good organic solvent, for example, tetrahydrofuran, into the aqueous media, it self-assembles instantaneously by itself. That will wrap the organic molecules to form the nanoparticle. And we can fine-tune the terminal group of the block copolymer so that we're able to produce nanoparticles with different functional groups and also different numbers of functional groups on the nanoparticle surface. So these functional groups will allow us to conduct reactions with recognition elements. It can be a peptide, it can be a DNA, it can be a protein, or even a small molecule, so that we're able to produce organic nanoparticles with the surface groups that shows high specificity for different kind of biological applications. Now, to make sure that every one of our students and researchers in the lab is able to produce the nanoparticles in high quality with uniform size distribution, so we went on to develop an automated system. This is really to avoid the batch-to-batch variation and people-to-people variation. So in this system, it contains 3 string pumps. The first and the third actually is a flow of aqueous media, and the central one is the organic media that contains the polymer at the wrap, and also the dye molecules or whatever reagents or drugs that you wanted to encapsulate into the nanoparticle. So we control the flow rates so that the mixing dynamics will be affected. And it's very interesting. If we drive this mixing to be very fast, for example, with the mixing time about 5 milliseconds, we're able to produce nanoparticles with a size about 20 nanometer. And if the mixing time is 10 milliseconds, the size will be 40 nanometer. And if the mixing is 15 milliseconds, the nanoparticle size can be managed at about 90 nanometer. So with this design, all we need to do is control the flow rate in the system. We're able to produce nanoparticles of very nice shape and different size, and more importantly, in large quantity, and it doesn't vary from time to time. So with a lot of high quality nanoparticles, it made us possible to explore all different kind of biological or biomedical applications. We also did detailed analysis for each of the nanoparticle. We wanted to understand, in terms of the organic nanoparticle versus the commercial quantum dots, what is the difference in terms of light emission, and what is the difference in terms of, for example, blinking, right? So what we find out was that for the same size of the nanoparticle, where we talk about the core shell with the overall size about 25 nanometer, right? So the photons emitted in 100 seconds for each of the organic nanoparticle are on average 10 to 40 times more than the commercial quantum dots. So that's the first time it gives us the confidence that organic nanoparticles actually can be made to be brighter than inorganic nanoparticles. And previously, we always thought that was not possible. Now, with this particular rate emissive AI molecules, we fabricate it into nanoparticles, and we find that this nanoparticle is very compatible with many equipments for different kind of applications. So it basically allows us to conduct in vivo experiments where we look at, for example, tumor locations in the mice. We can conduct cell experiments to visualize the cell structure, shape, cell interaction. We can also look at the tissue as well as the multi-photon imaging so that we're able to go deeper and with high resolution. And it's also interesting, the particle is suitable for super resolution imaging. So basically, it offers us a unique platform with the same nanoparticle, the same animal, where we can look at macroscopic to the nanoscopic structure to understand the disease of the tumor and all different kind of biological functions, which makes it very powerful strategy to study any disease or biological examples or questions of interest. Now, here I want to give you one specific example. What does that mean when we are able to produce organic nanoparticles that are brighter than, for example, commercial quantum dots and also the commercial dyes in terms of cell labeling? So this is a direct comparison of the nanoparticle labeled cells in different days when cell divides, how the fluorescence intensity is changing with the cell division across different time. And obviously, the AI nanoparticle labeled cells can last much longer. That gives very bright fluorescence, which allows us to track the cells over a period of time, for example, a couple of weeks or even a couple of months, depending on the different cells that we study. So how can we make use of this unique feature? I want to show you one short video where we take advantage of the zebrafish model. So here, this video shows how the individual cell divides until within a short period of time, in about a day or so, a cell is turned into a zebrafish. All right, now you pretty much see the zebrafish is already formed. It's a very lovely small species. And we like this, and we like this very much in a way because zebrafish, during the first lifespan, about the first 1 week or so, the yolk is able to supply all the energy and nutrition for the fish to grow, so that we don't need to feed the fish. We can fully utilize the fish as a model to study many interesting biological problems. Now, what we're particularly interested in is that there is a voice at the forehead of the fish. And once we open that, actually we can inject biological samples into that region to study how they interact with the fish tissue and circulation, etc. So this is exactly what we did. So we took the tissues from cancer patients. So some of the tissues are benign, and some of the tissues are cancerous, and some of the tissues can undergo cancer metastasis. And what we discovered was that dependent on the nature of the tissue and whether the cancer cells in whether the cells in the tissue is cancerous, non-cancerous, or can undergo cancer metastasis, it shows different pattern. So the top row here basically are the cancer patients with benign tissue. So the tissues actually will die after a day or two after it injects into the brain of the zebrafish. Now, these are the tissues from the cancerous patients, right, ovarian cancer. And these tissues, when it has been properly labeled with the AI nanoparticles and injected into the brain voids, they actually can survive and grow into a bigger mass. That's a clear indication of the cancerous nature of the cells inside the tissue. So basically, this allows us to visualize whether the tissue mass contains the cells that are cancerous versus the non-cancerous within a very short period of time, about a day or two. We will be able to clearly visualize the difference. And even more interesting is upon injection of the nanoparticle labeled tissue into the brain voids, and if we monitor the cancer cells, how they migrate across the cell, across the fish, it was found out when the tissue contains the cells that are not able to undergo cancer metastasis, they will just grow along the region that we inject the tissue. But if the cells can undergo cancer metastasis, actually we do observe that it can travel to long distance across the fish. And it's very interesting that each of the cancer cells is located next to the blood vessel, because cancer cells are really smart in a way they need nutrition all the time. So that's how we observe all this interesting phenomenon, and we further develop a mathematical model to monitor within a certain period of time what is the distance that the cancer cell travels, so that we're able to use that to quantify the degree of cancer metastasis. And the exciting thing about this is that everything is clear. We can visualize so that within about 48 hours, we can see through the whole process to understand the nature of the cells, whether it's cancerous, non-cancerous, versus whether it can undergo the cancer metastasis. Now, after this visualization, the key purpose is to use this model to do the drug screening. As we are able to actually inject the tissue that is directly taken out from the patient example, right? So this basically is a simple model that allows us to test the drug combination and the different drug doses. How does that can really help to inhibit the tumor growth? So from this combination, we will be able to recommend the drug and drug dosage that are highly tailored for individual patients. And we're in the process of working with the clinicians and researchers from our Cancer Institute to strengthen the model and make it generalized for practical applications. All right, so after we are able to fabricate the nanoparticles and utilize that for the imaging and cell tracing experiment, so very straightforward, naturally, we ask ourselves whether it's possible if we change the functions of the molecules inside of the nanoparticles so that we can further upgrade our nanoparticle from the sensing imaging reagent to the theranostic nanoparticles. And I would like to share with you two stories of our AI nanoparticles that can be used for the imaging and treatment applications. And alongside, we're also trying to solve the problems that are associated with the optical sensing, imaging, and therapy. One of the key issues is the penetration depth. Whenever we're looking at optical imaging or optical-based therapy, the penetration depth is very low, right? It's at the sub-millimeter. Maximum is millimeter depth, which actually in many cases is not really very practically applicable. So in this case, we developed one strategy, which we call the chemiluminescence guided cancer therapy. We wanted to eliminate the requirement of light excitation during the phototherapy. And what we did was to incorporate a chemical drug or a chemical which contains high energy. And we incorporate this chemical with a photosensitizer that we designed, and we form a nanoparticle. This nanoparticle will be responsive to hydrogen peroxide, which is a biomarker at the cancer site, and it will instantaneously gives out the single oxygen as well as the light emission. So the reaction mechanism is shown here. So this chemical, CPPO, in the presence of the hydrogen peroxide, will form this high energy intermediate species. And this molecule will interact with our photosensitizer and transfer the chemical energy to the photosensitizer and excite it to give out the light emission as well as the single oxygen production. So all these things will happen instantaneously as far as we're able to send the nanoparticle to the tumor site. It will react with hydrogen peroxide, and you see the light, and you see the treatment immediately. So these are the animal experiments where the animal actually had a tumor next to the left front arm. And if we look at the traditional fluorescence imaging, we will be able to identify the tumor site as well as the liver, because nanoparticles actually will have a broad distribution in the animal, and naturally it will accumulate a lot in the liver, so that we will have the tumor site clearly visible, right? Now, if we only look at the chemiluminescence, we will not be able to observe any signal that is outside of the tumor region, which make it highly specific. So in this case, we will only be able to observe the light emission from the tumor. And as far as we see the light emission, it is a signal to also show that the therapeutic function also starts instantaneously, because as the molecule has been excited, it gives out the light, it also produces the single oxygen at the same time. So that is why with these nanoparticles, we will be able to significantly inhibit the tumor growth, while for any other control experiments, the tumor actually will grow with the time. So that's a clear indication of how, through the molecular design and nanoparticle formulation, we are able to create smart solutions that is able to solve some of the treatment problems, and yet at the same time to eliminate the disadvantage associated with the traditional phototherapy that relies on the light illumination. In this case, we rely on the chemical reaction to actually supply the energy that's needed for the photosensitizer. All right, this is the last story. To solve the problem of poor selectivity between the drug for the cancers versus the normal tissue, right? So when patients actually has been diagnosed with cancer, very much likely there's a high chance that during the treatment, the large amount of the drugs that not only help to kill the cancer cell, but at the same time also kill a lot of normal cells, so that our patients become weaker and weaker over the time. And to prevent situations like this, we decided to look into the possibility of utilizing the microenvironment of the tumor to synthesize the drugs inside of the living system, so that it shows high selectivity to the tumor, but not to the normal cell or the health tissue. So in this case, we utilize a copper-based metal organic framework. So it's a porous materials with a copper at the node that links a lot of organic ligands. And we loaded two chemicals into the voids, the pores of this porous material. And we further wrap this material with a polymer shell so that we form a nanoparticle. Once this nanoparticle travels and reaches the cancer site, the biomarkers of the cancer, such as GFH, will cause the moth to decompose, so that the copper will be released as the copper 1. this copper 1 will serve as the catalyst to cause these two molecules to react in order to form a drug molecules inside of the cancer cell. So that shows high specificity to cancer cell because the same reaction doesn't occur if it's in a normal cell. So these are the two monomers that we selected in order to do the reaction. And the reaction is between the enzyme and the triple bond. It's a click reaction. And we, on purposely, also designed two positive charges in one part of this molecule is to make sure after this drug is synthesized inside of the cell, we hope that the drug molecule will also migrate into the mitochondria, which is the organelle inside of the cell that contains the DNAs, in order to make sure this drug is most effective in killing the cancer cell. So the cell imaging results is very promising. If we compare the MOF carrier, which carries the two precursors, versus the drug molecule that has been pre-synthesized before we use it to interact with the cell, actually it shows the same results. That's indicating it is effective for the two precursors to undergo the chemical reaction inside of the cell. And at the same time, we also overlap the imaging from our molecules with the commercial reagent, which is the mutual tracker, right? So they overlap very well. Right? Green gives you the yellow color, that indicating we're also very successful in sending the drug molecules into the mitochondria. And if we don't have the MOF as the carrier, the two monomers are not going to enter the cancer cells at the same time, and they also lack of the catalyst so that the reaction doesn't occur, right? So this only happens in HeLa cell, which is a cancer cell, because it's overexpressed the GSH. Now, on the right side is the normal cell. Normal cell doesn't have a high concentration of the GSH, and therefore we're not able to synthesize the molecules in situ. And that's how we can create a scenario to have the drugs for cancer cell, but no drugs for normal cell, even though we are actually shipping the same precursors and carrier into the cancer versus non-cancer cells. Now, to illustrate the therapeutic function, we rely on the zebrafish with the liver cancer, and the green color you see here is the green fluorescent protein. So basically, the poor fish had the liver cancer that's showing up in the green color, right? So if we don't have any treatment, of course, as the time goes by, the liver cancer becomes vigorous, and then the liver becomes bigger and bigger. The poor fish will die very soon. Now, if we use the meta-organic framework to incorporate the molecules and then inject into the fish, it's very nice that all the drugs are localized in the liver cells. And along with the treatment, you will see that the liver actually remains a small size and shape, right? That's much, much smaller as compared to that without treatment, indicating that the fish is in a good shape. Now, if we pre-synthesize the drug molecule, means we just directly inject the drug into the fish, then what we observe is this drug will spread out all over the fish body, while there are some parts that are inside of the liver. So that's exactly what we're trying to avoid. Whenever the animal or the patient takes the drug, we want the drug to go to the place to take effect rather than everywhere to show the side effect. So in this case, because this is photosensitizer, we have to make sure that this group of fish are staying away from the light illumination. Otherwise, there will be too many radicals formed to kill them even before the tumor has been treated. So there's all different sets of the control experiments, but nonetheless, it shows how important it is when we are able to precisely synthesize the drug in the tumor region so that it shows high selectivity in treatment as well as to eliminate all the side effects that has been general in many practices. All right, so with this, I would like to end the presentation. So today I talked about a special type of molecules with aggregation-induced emission. And from there, we develop a series of probes, such as the light-up probes, where we take advantage of the weak emission or very low emission of molecular probes for the light-up sensing and imaging. And then we talk about AIE dots, which are the AIE nanoparticles that we take advantage of the other side of the AIE property to offer high luminescence in the aggregated state. And then we talk about how we're able to integrate many design functions into the same molecule so that we can produce AIE photosensitizers and use that for image-guided therapy and image-guided surgery. And of course, we can also take advantage of bioosmolar reaction, which is the natural reaction that occurs in the living system, so that our AI molecules can be naturally incorporated into the cells or bacteria for many different kind of applications. So if you wanted to know more about our research, you can read some of the recent articles and also a few reviews that I attach here, so that you will be kind of exposed to more of the history and progress that we made over the past few years. All right, so I would like to thank my research team, and I'm really proud that our group has a large amount of female students. Great. And I would like to thank my collaborators as well as the funding agency. And I thank you very much for your attention. And now I'm ready to take any of your questions. Thank you.
谢谢刘教授。那跟在今天这个很短暂的1个小时，我以为会非常专业到我听不懂的状态，但是呢，刘教授深入浅出的用很多实验的案例来跟我们说明这么多的专有名词跟它相应的研究工作，那跟我们展示了用我不知道这样说对不对啊？是这个分子的结构跟特性来进行生物医学以及癌症细胞侦测，那甚至到这个 photosynthesizer， 我看这个中文应该叫光化学的动力疗法这样的原理，以及它怎么样运用在这个癌症的治疗上面。我觉得这些应用研究都非常的有意义。那也感受到了为什么刘教授可以做到今天这样子的一个学术的成就哈。那接下来呢，我们就欢迎我们所有的听众可以来跟刘教授直接的互动。那你可以使用这个举手的符号，也可以使用这个聊天板来分享你的想法或提问。那我来请祁宏，祁宏你要开视讯，然后跟刘教授互动吗？
Okay, yeah. So good morning, Professor. So I have 2 questions because user can be attached to AIE dye on the drug, and can be the diffusion of the drug on this cell inside. So my question is that because when this AIE attached on molecular surface, the drug molecular surface in theory, there will be changes in their molecular weights and change the surface charge. So will this kind of this AIE dye to effect for this drug layer metabolic in the cell inside, or to change their stabilize in the cytoplasm inside? So this is my first question. Thank you.
Okay, great question. It is true that when we attach an AIE dye to the drug, I'm quite sure it will change the behavior and even the location and its interaction with cells and everything, right? So that's why we have to be very careful in how we conjugate the AIE dyes to the drugs. We barely will do a fixed chemical reaction between the AIE molecule and the drug, but very often we actually do an active linker so that the AIE is attached to the drug, but once the drug reaches the region of interest, the AIE molecule should be detached. So it helps you to trace what's going on in the biological process. But once to the point that drug is taking effect, we hope that the AI molecule will be disconnected so that it will not affect the drug behavior, distribution, and everything. So in this case, the molecule will only serve as a tracing purpose. If they permanently coming together, it will affect the distribution and maybe also the biological function of your drug molecule. And we need to be very careful about that.
Okay, so my second question is because just like you said, we can use AIE and we can be target the peptide or glycan after this bacterial surface and we can be killed. So my question is because this is a targeting this bacterial, their cell wall. But when the white blood cell or the others, the soma cell, they eat this bacterial, so phagocytosis, they eat this bacterial and put this bacterial degree on the sodium hypochlorite, so the respiratory burst. Bacteria is dead, but our body, they cannot be degraded as a cell wall. So will this AIE also target this dead bacterial slice? So if that's possible, this image observed from this experiment inside, if that's possible, that might be a dead bacterial slide. So this is a second question. Thank you.
Okay, good. This one actually went beyond what we have been doing in the laboratory. So at the moment, we care a lot about how we can design the AI molecules that is able to detect the bacteria, which has already invaded into the cell. That's the first step. The second step is how after the invasion, we're able to kill the bacteria. That's the second step. But you care more about the third step. After we kill the bacteria, what's going to happen, right? Yeah, so at the moment, I don't have a clear answer to that because we didn't really study in that much of the detail. I'm hoping that cells themselves are smart enough to have a mechanism to clear out, let's say, a dead bacteria or a particle that it doesn't need, right? So what we have achieved is to make sure the bacteria will not do any harm anymore. So how it has been degraded or how it has been cleared out of the cell, we didn't study that yet.
Yeah, I remember something white blood cell can use exocytosis to clean out dead bacterial slides to the extracellular space. So when this AI also targeting, it's kind of going to the substance inside the slice.
Dependent on the molecular design of the AIE, its hydrophilicity, hydrophobicity, and charge, and also size, its location and interaction with the cells are very different. So we need to be specific to answer your question with a particular chemical structure. There's no general answer to that.
Okay, thank you, Professor.
那我们就可以把这个第三阶段的还没有研究到的部分呢，交给我们这个未来的科学家们哈，尤其是像祁宏这么优秀的这个，他只是一个高中生哈，那可以在自己的知识领域上面达到一个高度，我觉得这是非常了不起的。那顺着这个话题呢，我就想请刘教授，我们稍稍转向这个话题，就是您可不可以综合性的跟我们分享，就是说你在学习这个专业的这个路上，你什么时候开始对奈米科技产生兴趣？然后你怎么样进入到这个专业领域？每天花多少时间在实验室？那你怎么样走到今天这个地方？你综合性的跟我们分享你这一路上的一个经历，然后包括像祁宏他们这样子的学生怎么样，未来可能会面对什么这样
？Okay, good question. 我觉得是这样的啊，我觉得我是比较幸运的。为什么幸运呢？因为我当初去选择科研方向的时候，就选了一条比较特别的路。当时我记得我读那个PhD 的时候，我的研究方向是水溶性的共轭聚合物。那个时候大概在20年前的时候，当时聚合物非常的流行，因为2001年，2000年或者2001年那个诺贝尔奖是给了那个聚合物的3位科学家，2000年吧好像是，然后那个时候呢，大家的主要研究方向就是有机共轭聚合物怎么去做有机光电器件 OLED 我记得 PLED 是非常非常流行的那个时候。然后那个时候呢，就有一个问题不能解决，就是当这个器件 从单层发展到多层的时候，那个第一层的这个分子成膜了之后，第二层的分子如果再来成膜，如果他们用的是相同的或者相似的溶剂，那么下一层膜跟上一层膜就会有影响，那个膜的质量就不能很好的保证。所以那个时候大家就想说，可不可以第二次成膜的那个溶剂跟第一层的不一样，比如说第一层是甲苯或者是有机溶剂，那么第二层膜的时候，我们可以用水或者是甲醇这样的这个极性溶剂。所以当时呢，我就去研究这个课题，就是怎么去把有机的这个共轭聚合物从有机相要转移到水相。那个时候很艰苦，因为很少有人做这个事情，我就真的很艰苦，我做了很久都做不出来，直到后来也不知道哪一天为什么就可以做出来了。那我们就解决了一个问题，就是怎么把有机相的分子转移到这个水相。之后呢，就是因为这个独特的科研方向，使得我有机会去UCSB 去做 postdoc。那么做 postdoc 的时候呢，刚好那个时候 Alan Krueger 拿到这个共轭聚合物的这个诺贝尔奖，然后他有一个非常奇特的想法，他的想法就是这个有机的这个化合物已经做了这个器件，已经形成了一个特别的这样一个课题，可以拿到诺贝尔奖了。有没有可能性这一类的化合物从此以后去到这个生物的领域去做这个生物治疗这样的一个方向？然后那个时候是不可以想象的，因为没有人做，也没有人知道这个事情可不可以做。然后我记得那个时候就招了2个还是3个postdoc 就开始这个方向。然后我就是那个时候就从有机纳米链有机聚合物、有机纳米有机聚合物、有机光电开始转向这个生物，然后治疗，那个跨度很大，中间有一段时间真的也是很痛苦，因为天天在学不懂的东西。但是呢，这个学习的好处就是，当你学了好几个领域之后，突然有一天你就开窍了，你就发现说，原来当你游走于2个不同的领域中间的时候，其实你的机会就多了很多。因为你可以看到一个不同的视野，你可以看到很多不同的课题，然后你竞争的对手相对来说也会比较少。然后我就做完了 postdoc 就回到新加坡国立大学。那个时候我遇到一个很大的挑战，因为我回去的不是化学系，我回去的是化工系。我们化工系的学生跟化学系的学生不一样，化学系的学生喜欢做合成，化工系的学生喜欢做混合在一起比较大剂量比较大规模所以他们就不要去做那个分子合成。那么如果我不做分子合成的话，只有一条路可以走，那就是去做 formulation。formulation 的意思就像我们吃的那个胶囊，我们感冒了吃那个胶囊，对吗？胶囊有一个壳，那个壳里面放很多的那些药物。所以当时我是没有选择的选择，为了适应我的学生，我们就说好，那现在我们就改变从每一个分子的角度去改进这个分子，去看它的功能。我们可不可以找一个 general 的这个 strategy， 把所有的大家已经做成的千万个有机的分子，用一种 formulation 的方法把它们带到水相，那么我们也可以有很多东西可以研究。那就是那个时候我们花了蛮长的时间去研究这个纳米粒子的形成，怎么样的方法能形成好的纳米粒子，怎么样make sure 它们可重复，怎么样从人工到机械化的去生产这个纳米粒子。那之后呢我们就找到了很多新的科研方向，因为我们把所有的有机相的材料都拿到水相去看它们在水相的性质功能跟在有机相有没有什么不一样。因为研究的人很少，所以我们发现的东西就比较容易让大家接受。然后在这个基础上，我们就从成像，然后慢慢的看到说它可以治疗，然后慢慢的看到说如果我们把好多的这个功能的这个功能就是 集中到那个分子的设计上，那么它就能产生很多不同的应用。所以我们就相当于一步一步的把别人的优秀的成果吸收过来，然后我们又在自己的基础上一步一步的往前推进，所以就逐渐的形成了我们自己的体系。所以是一个学习的过程，是一个很激动人心的过程，但是也是一个很艰难的过程，因为我们通常都是在学我们不懂的东西。好像前年这个 COVID 来了，实验室关了，一个化学的实验室关了，就表示什么都没有了，然后我们就关了差不多2-3个月的时间。那怎么办呢？我就去跟我的同事学这个 modeling 和 machine learning。后来我们才发现说，真的是因为实验室关了，真的是因为我不可以去做实验了，我才去学这个东西。学了之后，我才再回过头来看说，哎呀，幸好关了，幸好我去学了，因为这个使得我们设计材料和选择材料的速度比以前快了很多。更重要的是，它让我们的成功率提高了很多，所以我们的学生变得更自信了，因为他做一个东西就有用，做一个东西就有他原来的性质，所以反而我们的效率提高了。所以有时候你真的不知道说发生了一件不好的事情，它产生的效果是负面的呢，还是正面的，完全看我们去怎么对待这样的一个事情。
这完全就是一个把危机化为转机的一个过程哈。那从这个过程里面，我们就听到说，成为一个杰出的研究者，要有很敏感的这个侦测潜力方向的这个敏感度。然后要很勇敢的投入，所以要鼓励同学们，就是碰到你陌生的，其实你不要害怕，对不对？就是要很勇敢的投入，即便我不知道，我都投入。然后一定会面对很多不懂的，然后失败的，还有困难的，你要一直坚持下去，就是那个 perseverance。然后在这个碰到跨领域的内容，或者是不同领域的人，然后不同的视角，要很开放的心态。也就是说，不要觉得我跟他不太一样。比如说像我举个例子来说，其实我主持这个盖亚的讲座，我们要面对很多不同的科学的领域，其实我自己也是不懂的，但是很勇敢的去接受它，然后在不同的场次里面去做学习。我觉得产生这样的一个视角跟这个过程，其实你会学到很多的东西。那所以我们也从刘彬老师的身上呢，就看到他怎么样去一直不断的产生新的研究方向，然后今天的成就这个突破呢，其实不是他的目标，但是是一个结果，因为这是在很多的累积之下所产生的一个结果。这样好，那因为今天时间的关系呢，那所以我们呢可能要先把我们今天的讲座到这边先告一个段落。那谢谢刘教授呢这么热忱的给我们同学们的回应以及鼓励。那我相信我们未来还有很多机会跟刘教授可以请教相关的问题，甚至再请刘教授跟我们分享下一个阶段您发现的这个新的结果是什么这样子。好，那我们也非常期待未来还一定有机会再跟刘彬仁教授来请教。我自己这边掌声，谢谢刘教授。那我一定会再跟您做更多的联络，谢谢，
谢谢大家，谢谢大家，
谢谢。